Today's lecture is going to take a rather detailed and in-depth look at the innate immune response. Briefly, we talked about an overview of the innate immune response at the end of the last lecture. What we discussed in that lecture was that, first of all, the innate immune response is in fact the first line of defense. It is non-specific in its mechanisms of recognition of pathogen. And this process does in fact rely very heavily on the process or mechanism of phagocytosis and phagocytes themselves. We also introduced two additional terms that are very important in relationship to the innate immune response. The first is PRRs, or pattern recognition receptors. These are receptor molecules, structures that are found typically associated with leukocytes, but can actually be secreted from leukocytes and do exactly what their name implies. They are pattern recognition receptors. They are molecular structures that interact and recognize pathogen-associated molecular patterns, or PAMPs. These pathogen-associated molecular patterns are components, structural characteristics, or also molecules that can be released from potential pathogens, things like lipids, carbohydrates, proteins, and in some instances, nucleic acid motifs can be recognized by PRRs. These are things that are broad, yet specific for a typical invading pathogen. They are broad, meaning that they cover pathogen categories and not necessarily specific pathogens like what we will discuss when we get to the adaptive immune response. We also introduced in the last lecture what are considered the functions of the innate immune system. Probably the most important component of the innate immune response is to identify and remove foreign substances that are causing a potential threat or can cause a potential threat. We also know that another function of the innate immune response is to recruit immune cells to the site of an infection. Additionally, the innate immune response plays a role in activating the adaptive immune response, as well as activating the complement system. So, as you can see, the innate immune response does collectively have numerous important functions in being the first line of defense against a potential pathogen. This slide is just another nice overview slide of what's happening in the innate immune response. Number one, there are physical and chemical barriers that need to be overcome in order for an infection to take place. We will discuss the physical and chemical barriers in just a little bit, but in order for an infection to take place, there needs to be a breach of this initial barrier of the potential pathogen being able to enter into the host system. Once that breach does in fact occur, there are a number of cellular responses that take place upon an infection occurring. And again, we will delve much more deeply throughout this lecture into these cellular responses that are occurring once the breach of the physical barrier has taken place. In addition to these cellular responses to infection, we know that part of the function of the innate immune response is to activate the adaptive immune response. And so everything collectively on this slide is what we are going to focus on in much more detail today. So the very first thing I mentioned on the previous slide was that there needs to be a breach of a physical barrier in order for an infection to take place. How exactly does that breach take place is what we are going to discuss with this particular slide and the next slide. The number one component that prevents potential pathogens from entering into the host system, again, is a physical barrier. Our external surface, our skin, allows for that protection from the external environment, keeping those potential pathogens from entering into the host system. We do know, however, that the skin also is involved in producing molecules known as antimicrobial peptides. We discussed or introduced antimicrobial peptides in previous lectures. These are small peptide fragments, meaning they are short stretches of amino acids that play a role in being antimicrobial, that is, somehow fighting off microbial infections. We will discuss these in more detail in a later lecture, including the number of categories that are associated with functions of antimicrobial peptides. However, most antimicrobial peptides actually play a role in inhibiting pathogen growth by blocking cell wall synthesis or somehow breaching that barrier overall. So again, we will discuss those in a little more detail a little bit later, but just for now, keep in mind that the skin, again, acts as a physical barrier, but in addition, those cells that are present there do in fact produce antimicrobial peptides that help fight off a potential pathogen from entering into the host system. But the skin alone is not the only thing protecting us from potential pathogens invading or entering into the host system. From this table, you can see lots of different examples of physical barriers or anatomical barriers that allow for a layer of protection. I'm going to highlight a few from this table. For example, our tears and our saliva do in fact contain enzymes known as proteases, and these proteases are a general class of enzymes that break down proteins. The job of these enzymes is to then potentially destroy antigen associated with pathogen. Additionally, tears and saliva produce antimicrobial peptides. They contain antimicrobial peptides, and these antimicrobial peptides, like we just mentioned, play a role in fighting off potential pathogens from entering in by disrupting the cellular or cell wall structure of the potential pathogen. When we discuss things like the stomach, we know that the stomach itself has a very low pH, and the job of that low pH is typically to help with the digestion of the food that has been ingested. However, that low pH also plays a really big role in destroying potential pathogens that may have been entering in through the process of eating or ingestion. Additionally, there are enzymes that play a role in the breakdown of potential pathogens, as well as antimicrobial peptides found in the stomach as well. The small intestine also contains enzymes and antimicrobial peptides that play a role in the destruction of potential pathogens. Another important feature I want to highlight is our respiratory tract, our airway and our lungs. There are cilia that are there that sweep mucus outward. Coughing and sneezing play a role in expelling that mucus, and that's that mucus that traps potential pathogens and helps the further removal of those potential pathogens from that site of potential entry. So again, to summarize from this particular table, you can see that there are lots of ways to protect the vertebrate system from pathogens actually entering into the system. So this is again truly the first line of defense, physical and chemical barriers that prevent potential pathogens from entering into the host system. Now, before I focus on the content of this particular slide, I want to provide you with some specific examples of molecules that are involved in protecting the host from an infection actually taking place. The first one is an enzyme known as lysozyme. Lysozyme is found in mucosal secretions, tears, saliva, respiratory tract. What this enzyme does is cleave peptidoglycan that is found in the cell walls of bacterial pathogens and also some fungal pathogens. This cleaving of the peptidoglycan leads to the lysis of the cell, meaning the lysis of the potential pathogen, and destroys it before it actually enters into the host system. The next protein or molecule I want to talk about is a protein known as psoriasin. Psoriasin is found in the skin as well as in mucosal secretions, like we mentioned previously, tears, saliva, respiratory tract, intestinal tract, urogenital tract, anywhere there is a mucosal lining. What this particular protein does is disrupt membranes associated with potential pathogens. And by disrupting the membrane, again, that will cause destruction of that potential pathogen. The last specific example I want to give you are proteins known collectively as surfactant proteins. These are typically found in the respiratory tract, in mucosal linings, and the job of the surfactant protein is to block surface components of the potential pathogen, but more importantly, what this does is promote phagocytosis of that pathogen. So again, some specific examples of molecules that are playing a role in protecting the vertebrate system before an infection actually takes place. This particular slide just drives home this concept that there is a layer of protection, in particular chemicals that can be produced or molecules that are produced that help protect the host from a potential infection. In this particular example, we are looking at the epithelial layers that are producing protective molecules to again protect from a potential infection. As listed here, these would be things like an acidic pH, enzymes and binding proteins, and antimicrobial peptides. So what I want to highlight is what's seen in the figure on the right-hand side. In this experiment, what was done was 2 different types of bacteria were utilized to inoculate the skin of the test subject. The first one is Staphylococcus aureus, which is not necessarily always a pathogen, but in some cases can cause skin infections, respiratory infections, and also can play a role in food poisoning. In addition, the second bacterium is E. coli or Escherichia coli, which in most cases is a non-virulent type of bacterium. Now, in this experiment, the individual was inoculated with either Staphylococcus aureus or Escherichia coli, and a 30-minute incubation period was taken place. Following that 30-minute incubation period, the individual was instructed to place their fingers where they were coming in contact with either forms of the bacteria onto a plate that contains nutrients and a substance that allows for bacteria to be grown and cultured. Those plates were then incubated, and what you can see is that the Escherichia coli containing plate did not grow any E. coli, whereas the Staphylococcus aureus did grow. This clearly shows that there's a differentiation between the types of bacteria that can be recognized by molecules or chemicals that are produced by the skin, and clearly E. coli has been destroyed, whereas Staphylococcus aureus was not destroyed. So there is a differential between the level of protection, so it's not straight across the board that every single bacterium is going to be destroyed before entering into the host system, but this clearly demonstrates that there are in fact components playing a role in destroying potential pathogens. Okay, so let's review one more time what the functions of the innate immune system or the innate immune response is. It is to recruit immune cells to the site of an infection. It is to identify and remove foreign substances that are a potential threat, and to activate both the adaptive immune response as well as activate the complement system. Key components that are playing a role in this mechanism are PRRs, which are pattern recognition receptors. These recognize patterns, broad structures, or motifs that are highly conserved but absent in the host, highly conserved in a potential pathogen and absent in the host. These pattern recognition receptors mostly are membrane-bound receptor molecules. They can be cytoplasmic. They can also be secreted, and we will look at some specific PRRs later on in this lecture. Some PRRs can act as opsonins, and we discussed absonins in the last lecture, as these are molecules that target an antigen for phagocytosis. The pattern recognition receptors recognize broad patterns in pathogens, and upon this recognition, there is a release of cytotoxic molecules, there's a release of cytokines. This is allowing for the induction of phagocytosis and activation of both complement and the adaptive immune response. Now, like I mentioned just a few slides ago, the very first thing that has to happen in order for an infection to take place is a breach of a physical barrier. Once the breach of the physical barrier has occurred, the first steps of the innate immune response is inflammation. So inflammation is induced. It is a localized tissue response that includes or causes pain, heat, redness, and swelling. These are the key characteristics of inflammation, and they are due to a number of physiological things that are occurring as part of the inflammatory response. The pain, heat, redness, and swelling is due to altered blood flow at the localized tissue region. There is an influx of immune cells that occurs in response to inflammation. Removal of the foreign antigens occurs. And lastly, healing of damaged tissues take place. So with inflammation, what happens within the minutes of some type of tissue injury and detection of a potential pathogen, there is an increase in the vascular diameter, which is known as vasodilation, and this allows that increased blood volume or increased blood flow to the target area. This increased blood flow to the target area is what's going to allow for additional immune cells to begin to fight this potential infection. So let's look at this step by step. The first thing, as I just mentioned, that needs to occur in order for an infection to take place is the breach of a physical barrier. That breach of the physical barrier in this particular slide is demonstrated by a blade cutting into the host epithelial layer, and it is this cut that causes tissue damage and allows for potential pathogens to now enter into the host system. This is where that breach has taken place. Now, when this breach has taken place, the cells that are being damaged can produce molecules known as cytokines. Cytokines, as I introduced in the last lecture, are signaling molecules that help with the immune response. So, cells that have been damaged due to physical force or physical trauma allow for the secretion, production, and secretion of cytokines. The class of cytokines that are typically produced by this damage are referred to as being pro-inflammatory. That is, they promote the inflammatory response. I'm going to come back to what some of those cytokines do in just a minute. But at the same time, there is this breach of the physical barrier, and those damaged cells produce cytokines. There are always immune cells found within the tissues that are searching, hunting, looking for potential pathogens that may have entered into the host system. The cell types that are typically involved in this continual search of potential pathogens are usually neutrophils, but can include dendritic cells, can also include a few other cell types, but neutrophils and dendritic cells are the number 1 and number 2 types of cells that are typically doing this surveillance, this searching for potential pathogens. These leukocytes have PRRs, or pattern recognition receptors, that can bind and recognize PAMPs, pathogen-associated molecular patterns, on these bacteria or other pathogens that may have entered in after the physical breach. So what does the interaction between the PRR on the leukocyte and the PAMP that is part of the pathogen, what actually happens when that interaction takes place? Typically, it triggers phagocytosis, and we're going to look at phagocytosis in more detail on the next slide. But in addition to that, it also allows for additional cytokine production from the leukocytes. So not only do we have the damaged cells producing cytokines, but these leukocytes that have now done the first interaction with a pathogen also are secreting cytokines. Collectively, these are pro-inflammatory cytokines and do a number of things. They promote the inflammatory response. They promote the inflammatory response by sending a chemical signal to allow for vascular permeability, increased blood flow to the target location, and to help summon or call additional leukocytes to the target location. These molecules allow for leukocytes that are traveling in the blood to receive the signal and then exit the bloodstream and infiltrate the site where the potential infection has taken place. So the cytokines that have been produced or secreted include general molecules that are helping in this inflammatory response, but there are a subset of cytokines known specifically as chemokines that are molecular attractants. Their job is to play a role in attracting those additional leukocytes to the target location. So before we look at phagocytosis in a little more detail, let's briefly discuss some of the key cell types that are involved in this inflammatory response, part of the innate immune response. Neutrophils, as we discussed in the previous lecture, are the most abundant of the leukocytes. They are, in fact, phagocytes. They produce reactive oxygen and reactive nitrogen species. They also produce antimicrobial peptides. These are typically the first responders in the innate immune response because of their abundance in the overall system. They are typically the first to migrate from the blood to the site of an infection as well. In addition to neutrophils, macrophages do play a really big role in this process. They are also phagocytes. They produce cytokines. They produce antimicrobial peptides, reactive oxygen, and reactive nitrogen species. So very similar overall to the function of neutrophils. Additionally, dendritic cells play a really important role, doing some of the same things we just discussed, producing reactive oxygen and reactive nitrogen species, producing cytokines that play a role in the inflammatory response. They are phagocytes, and they also produce antimicrobial peptides. So these are probably the three key types of players that are involved in the initial detection of a potential pathogen and destruction then of those potential pathogens. Another cell type that we discussed when we discussed all of the cells previously are natural killer cells. Interestingly, natural killer cells do play a role in the innate immune response. They are really important in the defense against viral infection. They produce cytokines, and they induce apoptosis, which is a mechanism of programmed cell death that is going to destroy cells that have been infected with a virus. And so these do play a role in the innate immune response. The main process or the main component that's playing a role in the destruction of potential pathogens is the mechanism known as phagocytosis. Phagocytosis is an engulfment and internalization of materials such as microbes for their clearance and destruction. And there are a very clear set of distinct steps associated with phagocytosis. The first one is the recognition of the potential pathogen. Now, this recognition occurs due to PRRs, or pattern recognition receptors, that are associated with the membrane of the phagocyte. It is the PRRs that bind to PAMPs, pathogen-associated molecular patterns, that are found in association with the pathogen. This interaction between those two components allows for the recognition that we've been discussing as so important to occur. Once that recognition takes place, that then triggers the phagocytic process. The plasma membrane pinches in of the phagocyte, with the pathogen being taken in as part of that pinching in. The bacterium is ingested, forming what is known as a phagosome. This vesicle inside the phagocyte now contains the pathogen inside. The phagosome fuses with structural components, organelles known as lysosomes, and lysosomes contain hydrolytic enzymes or digestive enzymes that play a role in destroying that potential pathogen, breaking it down. Additionally, the pH of the inside of the phagosome, which is now referred to as a phagolysosome, is rather low, which helps with the destruction of that pathogen as well. Typically what happens is those digested components are then released from the cell, and those digested fragments are then filtered out of the host system for complete renewal or removal. Additionally, when we mentioned phagocytosis in a previous lecture, there is another possible fate of those fragments that have been produced by phagocytosis. Those fragments can be utilized as a link to the adaptive immune response, and we will discuss that again when we get to the adaptive immune response. But it's really important to understand that phagocytosis not only destroys the pathogen, but plays this essential role in linking the innate immune response with the adaptive immune response. So looking at inflammation or the inflammatory response in a little more detail, let's briefly review that it is an induced localized tissue response. It includes pain, heat, redness, and swelling at the localized area. What happens in this process is that there's an altered blood flow that takes place, there is an influx of immune cells, there is removal of foreign antigens, and then lastly there is healing of damaged tissues. This inflammatory response or inflammation occurs within a few hours of a potential infection. Leukocytes are recruited to the site of the infection in a process known as extravasation or extravasation. So what we're going to look at here in a little more detail is how those leukocytes exit the bloodstream and enter into the target location. So this is the process of those cells that are in the bloodstream receiving the signal that we talked about being produced by those initial leukocytes that interact with the pathogen. That signal is received and allows for the leukocytes that are traveling in the blood to exit the bloodstream and enter into the site of the infection. This process is highly regulated and is multi-step. The migration of the leukocytes from the bloodstream to the target site of the infection, again, is a crucial one taking place. Some of the cytokines that are produced that are pro-inflammatory promote vasodilation. Vasodilation simply means that there's an increase in the vascular diameter, which then allows for vascular permeability, allows for cells to exit the bloodstream and enter into the target location because the diameter of the blood vessel has actually gotten larger. The first step in extravasation is actually not part of the extravasation process. The first step is what's known as rolling. So leukocytes travel through the bloodstream with a mechanism simply referred to as rolling. These leukocytes attach very loosely to the endothelial cells that are part of the blood vessel, and they attach themselves via a family of proteins known as cell adhesion molecules, or CAMs. Cell adhesion molecules are found in the endothelial cells as well as in the leukocytes. These are molecules that allow for this interaction to take place between the leukocyte and the endothelial cell. And typically, leukocytes again move via this rolling mechanism. There's a continual breaking and binding, or binding and breaking, of the interaction between the cell adhesion molecules. So the pressure that is produced by the heart pumping and the blood circulating is what causes the breaking of that interaction, hence the movement of the cell in the bloodstream. What happens next during the process of inflammation is that there is an activation step by a cytokine, specifically referred to as a chemokine. So a chemokine is a chemoattractant. It interacts with the leukocyte and activates it to trigger a transformation in that leukocyte, which is the first signal to get the leukocyte out of the bloodstream and into the target site where the infection is occurring. This initial interaction or activation mechanism is due to a change in conformation of a specific cell adhesion molecule known as integrin. Integrin undergoes a conformation change, and when it undergoes the conformation change, allows for it to interact much more tightly with its corresponding cell adhesion molecule that is part of the endothelial cell. This tight interaction leads to step 3, which is arrest and adhesion. So there's this strong interaction that takes place between these molecules, and we'll look at them in more detail on the next slide. But that essentially anchors that cell at the position and doesn't allow it to continue to roll through the bloodstream when the heart pumps. The last step is transendothelial migration, and you can see that in step 4. Essentially what is happening is the cell is passing between the endothelial cells, that opening that has been generated because of the vasodilation that has taken place, allows for these cells to slip through the bloodstream and enter into the target location. So let's take a closer look at the molecules that are playing a role in the adhesion and the movement of the leukocyte from the bloodstream into the target area where an infection has taken place. The CAMs, or cell adhesion molecules that are important in this interaction, in the initial interaction, are mucins, which are part of neutrophils typically, and selectins, which are part of the endothelial cells. So these are two specific types of CAMs that interact with one another to allow for the cell adhesion to take place in the typical rolling mechanism. Once the chemical signal or the chemokine has been received, that chemical signal alters the structure of integrin, and you can see integrin in the pinkish color overall, and its structure altered when the signal has been received. And when that interaction or that change in conformation occurs, it allows for integrin to now bind very tightly with yet another endothelial cell adhesion molecule known as ICAM. And this is a specific cell adhesion molecule that binds with integrin, and this causes that anchoring or that strong adhesion to prevent the cell from continuing to roll and eventually exit the bloodstream at the target location. So what we've just done is discuss the cells and the cell types that are involved in the inflammatory response. But what I want to do now is focus a little bit more on the molecules that are involved in the inflammatory response. We've just discussed or spent a great deal of time discussing cytokines that are referred to as pro-inflammatory cytokines. Their job is to promote the inflammatory response or inflammation. There are over 200 different cytokines that have been identified with specialized function, some overlapping function, and we can spend an entire lecture or more discussing individual cytokines. We're not going to do that. We're going to look at 2 specific cytokines just so that you get some idea of what their function is or are. The first one is known as IL-1, which stands for interleukin-1. Interleukin-1 is actually a family of cytokines that contains 11 distinct members. The job of this particular cytokine is to help with increasing the expression of adhesion factors, those cell adhesion molecules that we just talked about, The interleukin-1 family of proteins is produced mainly by macrophages and dendritic cells, as well as epithelial cells, again, that we talked about having that damage occur allows for the production of certain cytokines, and this is one of them. Interleukin-1 also plays a role in vasodilation and plays a role in regulating the hypothalamus. The hypothalamus helps control body temperature, and with interleukin-1, it increases the body temperature, which then increases metabolic activity and hopefully and potentially inhibits the growth of some bacteria. The second cytokine I want to talk about is TNF-alpha, which stands for tumor necrosis factor alpha. It is a cytokine that promotes vascular permeability. It's produced mainly by macrophages and dendritic cells, like we just mentioned, but also helper T cells and natural killer cells are known to produce this particular cytokine as well. It also plays a role in promoting adhesion with those cell adhesion molecules that we discussed. It stimulates macrophage development, also stimulates the phagocytic activity. So you can see that there's some overlapping function with these two cytokines, but that's what a lot of cytokines do, is overlap in function. And again, I just wanted to highlight two specific cytokines for you. I've also mentioned a few times antimicrobial peptides, which are abbreviated AMPs. These antimicrobial peptides have been isolated from all types of organisms: humans, frogs, flies, worms, plants. So lots of organisms are known to produce antimicrobial peptides. In fact, there have been over 800 distinct antimicrobial peptides that have been identified from distinct and diverse species. This indicates that these particular molecules are pretty effective in their role in immune response. Antimicrobial peptides are short stretches of amino acids, typically anywhere from 6 to 59 amino acids long. They are produced by a variety of different cells, like we discussed with the leukocytes, but we also know that epithelial cells can produce them as well. The antimicrobial peptides do in fact tend to be fast-acting, meaning that they elicit an effect in about 90 minutes of exposure. I've mentioned before, but most antimicrobial peptides somehow disrupt microbial membranes or cell wall structure. There are other antimicrobial peptides, though, that are known to play a role in inhibiting DNA, RNA, and protein synthesis, and some have been identified that target viruses as well. Lastly, I want to talk about molecules that are collectively referred to as acute phase response proteins. These are proteins that got their name collectively because there was a dramatic change in their concentration in response to a typical infection during an inflammatory situation. So these are just molecules that have been noted to increase in their concentration dramatically in response to inflammation. We have a few that are listed here specifically. We have complement proteins, which are part of the complement system, that third facet that we will eventually get to. It turns out that these are produced in high abundance as part of the innate immune response. And again, one of the functions of the innate immune response is to activate complement. So it's not a surprise that the complement proteins actually go up in concentration because of that activation that we've talked about. Additionally, there are two other proteins listed here. There's C-reactive protein, known as CRP. This protein binds to polysaccharides on some microbes and does in fact promote phagocytosis. It is synthesized by the liver and is synthesized in response to cytokine production. Another one is mannose-binding lectin. This actually recognizes mannose, which is a specific pattern associated with many microbial cell walls, and this too acts to trigger phagocytosis overall. Okay, so what exactly happens when we have receptor-ligand interaction? I'm going to keep it as general as possible, but I think it's very important to understand that when receptor and ligand interact, it's not just magic. There is something specific that's occurring that allows the cell to behave in a specific way. So in this slide, on the top portion of the figure, you see a receptor, in this case a membrane-associated receptor, and a ligand, represented by the orange sphere, binding and interacting. Once that interaction takes place between the receptor and the ligand, typically what happens is that causes a signal to occur inside the cell and the cell to act in a specific way. Usually that signal involves conformation change of components that can bind to other molecules that are inside the cell. So in this top portion of the panel, you see this complex interaction that's taking place that is allowed to take place because of the receptor-ligand interaction in the first place. Oftentimes what happens in this complex interaction is this then causes a cascade of events to take place inside of the cell known as a signal transduction cascade, which then allows for signaling to occur inside the cell. So receptor-ligand interaction, conformation change on the interior portion of the receptor, which then allows for other molecules to interact and eventually allow for activation of other molecules inside the cell. The most common end result with the receptor-ligand interaction is the activation of a protein that is usually referred to as a transcription factor. A transcription factor is one that allows for transcription of genes to take place or occur once it has been activated. So this slide really nicely shows the difference between the inactive form in the red and the active form in the green. What has allowed for the activation is due to that signal transduction that we just discussed on the previous slide with the interaction of the receptor and the ligand. This ultimately leads to the activation of a protein, and that activation then again has some subsequent effect inside the cell. Like I just said, in most cases, this is a transcription factor, which allows for the transcription of genes involved in whatever this signal happens to be. Now, there are lots and lots and lots of different types of PRRs that are found within a typical leukocyte. And because there are so many of them, oftentimes what we like to do is try to categorize them or classify them in some way. The two ways that PRRs can be categorized are either by their function, like you see at the top, meaning that they are involved in some type of signaling event for A, or B, involved in endocytic events, which means leading to phagocytosis. So either the PRR allows for a signal to be received and initiated in the cell, or it triggers phagocytosis. And in some cases, it actually does both. Alternatively, PRRs can be classified or categorized by their cellular location. They can be membrane-bound, either on the cellular surface or a membrane inside the cell. They can be cytoplasmic, totally found within inside the cell, or they can be secreted from the cell, like we talked about in the previous slide, where the PRRs are being secreted and acting as opsonins. So what you see here are some specific examples of PRRs, those that are membrane-bound, those that are cytoplasmic, and those that are secreted. So here I want to give you a little more detailed example of a PAMP, which is a pathogen-associated molecular pattern that can be recognized by a PRR. In this particular figure, what you're looking at is E. coli, and E. coli happens to be a gram-negative type of bacterium, which just means it has a rather complex double membrane system and a peptidyl glycan layer that is in between the double membranes. More importantly, though, many gram-negative bacteria also have something known as a lipopolysaccharide. This is a molecular structure that is on the exterior surface, and this in fact is a type of PAMP, pathogen-associated molecular pattern. There are actually a number of different and distinct types of PRRs that specifically bind to this lipopolysaccharide. And so this just shows you again a little bit more detailed information of a PAMP, a pathogen-associated molecular pattern that can be recognized by a PRR. So what we're going to do now is look at some specific PRRs, pattern recognition receptors, and look at some characteristics and features associated with these specific PRRs. The first one, which is a very common type of PRR, is known as the toll-like receptor. These are transmembrane signal receptor proteins. In most cases, they signal phagocytic activity. They do signal production of cytokines. There is a family of these receptors. 11 of them have been identified in humans. A total of 13 have been identified in mammals in general. They typically induce the inflammatory response, and what you're looking at here is the common structural characteristic of a typical toll-like receptor. Now, as I just mentioned on the previous slide, the toll-like receptors actually are represented by a family of distinct receptors. My point here is to not have you memorize each one of the individual toll-like receptors, but just to exemplify the differences and the distinction found in individual toll-like receptors. What you see on the outer component associated with the yellow membrane are plasma or cell membrane associated toll-like receptors. And what you see is that they do in fact interact with lots of different types of pathogens. The first one, the toll-like receptor 4, recognizes gram-negative bacteria. The next one recognizes bacteria and parasites. The next one recognizes gram-positive bacteria and fungi. And the list goes on. So you can see that there's quite a bit of diversity in what is being recognized by individual toll-like receptors. But what I wanted to highlight with this particular slide is that there are also toll-like receptors that are found on the inside of a host cell. These are associated with endosome membranes or lysosome membranes, and particular what I want you to focus on is what these toll-like receptors are recognizing. They are recognizing viral components, viral proteins, viral RNA, in some cases viral DNA. And so, like we mentioned previously, the importance of having a receptor inside the cell is specifically to detect viral infections, because again, a virus has to get inside the host in order for the infection to take place. So what you see here is, again, this vast diversity with this one family of receptors. So the next type of receptor that I want to talk about in a little more detail is the C-type lectin receptor. The job of this group of receptors is to activate the innate inflammatory response. It is mostly involved in signaling pathways, and I'm going to get to that in just a minute. We see these particular receptors typically associated with dendritic cells and macrophages, but that doesn't mean it's not found in association with other leukocytes. Generally, this receptor recognizes a specific carbohydrate component found in some fungi, certain bacteria, as well as parasites. Because this receptor initiates a signaling pathway inside of the host cell, this is a really nice example that I was alluding to in the more generic example previously between receptor-ligand interaction. What I mentioned with that particular slide is that oftentimes when you have the receptor-ligand interaction, it activates transcription factor or transcription factors that play a role in allowing for transcription of specific genes to occur. and this is definitely one of those examples. All of the components in green are in fact transcription factors that have been activated by this interaction between the receptor and the ligand. And the transcription factors then allow for transcription of genes that code for cytokines, more specifically chemokines, and antimicrobial peptide sequences. So again, you can see that transcription is activated of specific components due to this type of interaction. The next specific receptor I wanted to focus on is known as NLR, nucleotide oligomerization domain receptors. These are activated by PAMPs and another category of molecules known as DAMPs, which are damage-associated molecular patterns. This particular type of receptor is found within an endosome, so this is something found inside of the cell. It is involved in signaling pathways as well. It is, in fact, highly conserved, can be found in lots of different types of organisms, including plants. It does activate pro-inflammatory cytokine production and antimicrobial protein production. These particular receptors typically recognize cell wall components, but can recognize some other features as well. But here too, what you see in green is the activated transcription factor, which then allows for the transcription of genes that code for cytokines, chemokines, and antimicrobial peptides as well. The last specific receptor, or PRR, that I want to talk about are what are known as the RIG-I-like receptors. This is short for retinoic acid inducible gene I-like receptors, hence easier to just call them RIG-I-like receptors. These are in fact receptors that are intracellular, meaning they are found inside of the cell overall. These are extremely important for viral detection. But yet again, what's highlighted here is that what happens when the signal is in fact received is that transcription factors are activated and allow for transcription of certain genes to take place, including genes that code for cytokines, specific chemokines, and antimicrobial peptides. Even though this last portion of the lecture has focused on receptor-ligand interactions in general, we have looked at more specifically receptor-ligand interactions between PRRs and PAMPs. But that receptor-ligand interaction is still a very important one. And in this particular slide, what are we looking at is the receptor, which is a receptor for a cytokine. So we've discussed that cytokines get produced and they somehow play a role in perpetuating or allowing for the immune response to continue. How do they do that? They are signals that have to be received by cells to act in a specific way. So the whole process of receptor-ligand interaction and the fate of that interaction and what happens to the cell once the interaction takes place is a similar concept like we just focused on. But again, this slide specifically refers to a receptor that is the receptor for a cytokine. So up at the very top, you have your plasma membrane in yellow, you have the receptor in the reddish-orange color, and then you have the ligand that's binding to the receptor in the green. In this case, the ligand is a cytokine that has been produced, and the receptor is the receptor for that particular cytokine. There is still an interaction that occurs on the interior surface of that particular receptor that then causes a signal transduction cascade to take place. And in this case, when that cytokine binds, it causes lots of different effects in the cell overall. Right at the center, what you can see, a similar concept we focused on just a few slides ago, is that transcription of certain genes is turned on, and so genes are allowed to be produced that are important for the cell to function or behave in a specific way. And the very last thought I want to leave you with is what's referred to as the ubiquity of the innate immune response. So this table has a lot of information in it, but what I highlighted in the yellow is the first column has different taxonomic groups, different types of species associated with them, and then whether we find the innate immune response present or the adaptive immune response or both. And so what I want to highlight is that the innate immune response is found in every organism that's listed on this table. So the innate immune response is a very general immune response found with lots of different types of organisms. Not just humans, not just vertebrates. You can even see that plants have an innate immune response as well. Contrast that to the adaptive immune response, which is definitely something that is vertebrate specific. Again, we haven't talked about the adaptive immune response. That'll be coming in the next unit of material, but I just wanted to highlight that for you, that the innate immune response is something that is found in lots of different types of species, very diverse types of species, whereas the adaptive immune response tends to be something that is vertebrate specific. And with that, we will end our discussion of the innate immune response.